And here we are at the end of the road. Now that you know everything you need to know to get beautiful data, we're going to talk about how to get it published. Let's take a look. Welcome back to our Introduction to Flow Cytometry series. If you've missed any of the previous episodes, you'll find a link to the playlist in the description. So far, we've covered how a cytometer works and the things you need to put together to have a good experiment, controls, understanding compensation, etc. Today, we're going to look at how you can take your data and make it publication quality. When we talk about publishing flow cytometry data, there are three main areas to consider. First, your experimental and sample information. Second, how you acquired your data. And third, your data analysis and presentation. Each category is important for accurate reporting in order to increase reproducibility and rigor, especially with your flow cytometry experiments. It's important to remember that the overriding aim of publishing your data is to provide enough methodological information so that somebody can reproduce your experiment. Failure to do so can seriously compromise the scientific conclusions reached in your experiment. So first up, experimental and sample preparation. This is really just a detailed description of your experimental design and your sample preparation, normally what you'd put in your materials and methods. However, generally in flow cytometry papers, this is quite lacking and needs a lot more detail. So when you're writing up this part, make sure you include details on how your samples are prepared. So this includes the number of independent experiments and the number of samples, whether you've done them in duplicate or triplicate, details on how the samples were prepared, so things like the buffer you used, the cell concentration, proteases if you added them, if you filtered your sample, if you fixed your samples and what reagents you used, if you permeabilized, what reagents you used. So anything that touches your cells, you should have enough detail in the buffer information in your methods that somebody could go and make that exact buffer. The same applies for your fluorescence reagents. Most papers you'll see will just list things like, we used CD3 FITC. However, this doesn't give somebody enough information to go and reproduce your experiment. So you should also make sure you include things like the clone you used, the vendor, and the catalog number, so if somebody wanted, they could go back and buy that same antibody. Generally, the easiest way to present this is in a table, like the one shown here. Next up is data acquisition. You might find in a lot of papers you read that this area will be severely lacking. Most of the time, you will see just the name of an instrument given, or it might just say, these samples were acquired on a flow cytometer. This does not give you enough information to go back and reproduce this. Again, the optical configuration of an instrument will have a large bearing on how that data can be interpreted. So it's important that when you're reading a paper, you not only know what instrument it was acquired on, but also the lasers and the filters that were used, as this can greatly determine the efficiency of the signal capture. Under data acquisition, this is also where you would discuss how you did your compensation. In this section, you would supply details on what you used for your compensation reagent, i.e. cells or beads, how your compensation was done, how your compensation was calculated, and in some cases you may even include your compensation matrix. Including this is important because uncompensated or improperly compensated experiments can result in artifacts in measurement which will change the quantification. And so for a reviewer or a reader reading your paper, it's important for them to understand that your compensation was done properly so that they can actually assess the findings that you are presenting. And last up for data acquisition is talking about the sample size and number of events acquired. This is important so that a reader can determine if enough events were acquired so that you can have a precise measurement of the population you're looking for. So for example, if you're looking for a cell that is found about 1 in every million cells, you'd want to make sure you have enough events acquired to account for that. So if you acquired only 1 million cells, you might find 1. That would not really be believable. If you acquired 100 million cells, you'd have 100 potentially. And so knowing that will allow a reader to assess how robust your data is, and then be able to draw their own conclusions from your data. So it's very important that they have those numbers. And finally, data analysis. This is the part that really draws the attention because this is where you start presenting your data. Now, like the other sections, a lot of the times you may find this lacking in detail to be able to assess what's truly happening on the plot. So your publications don't fall victim to this. A few things I would recommend adding to your plots all the time. First and foremost, always make sure you have your axes labeled. Now these labels shouldn't just be the antigen, they should also be the antigen, fluorochrome, and ideally also the filter used to collect the signal. Next up, do not remove the numbers and the scaling. This is critical. Understanding where your populations are scaled on a plot is important to interpreting the data. So don't remove the numbers. This will also allow people to see if your scale is log or linear, which again is important for interpreting the data. When dealing with your gates, it's important to give a gate percentage on these plots so that people know where you're getting your percentages from when you start presenting your data in more graphical formats. Similar to your data acquisition, it's also a good idea to put the number of events on your plot, especially if you're using contour plots. When using a contour plot, it may look like there's a lot of data on that plot, but that data may only be representative of a few data points. So if a person knows that there's 10,000 events on this plot versus 100 events on this plot, it can greatly affect how they interpret that data. The plot to the side now will show a pretty standard publication quality plot. You'll probably find plots like this in a lot of papers. Now, if you look at this plot, can you truly interpret what's going on in this experiment? Or what details do you wish you had? If this was me, I would say we're missing quite a few things. My first issue, I have no idea if the scaling on this plot is linear or logarithmic. There are no numbers, there is no scale. I have no idea how they are looking at their fluorescence. Second up, there's no fluorochromes listed. Also, no gate statistics, so I have no idea what percentage is represented on this plot. And finally, there's no cell number given for the number of events on this plot, so I don't know how representative or believable these populations are. So when putting your plots together, make sure these details are all included and you don't fall into this trap. In most manuscripts, you'll also be asked to include a gating strategy. This is essentially just a walkthrough of all the gates you used in your experiment to get from your initial collection of data to your populations that you analyze in the end. Basically, the step-by-step walkthrough of your gating hierarchy. Now, this is really nice to include because it shows people the logic that you use to get through your populations, but it also allows readers to assess your gating strategy. This is generally included in the supplementary data, as for the most part in the main body of the manuscript, you'll include the final figure. Important things to have in your gating strategy include things like how you discriminated doublets, if you used a time gate to exclude problem areas, any FMOs you used, etc. So this is where you showcase and highlight all your controls and how you applied them to your experiment. A representative gating strategy is shown here. Now don't worry about the details, but things that I want to highlight are the use of arrows to move from population to population, and how they showed the FMO versus the samples so that people can very visually see how those FMO controls were applied. Lucky for you, all of this is compiled in one easy document known as My Flow Cyt, or the minimum information for a flow cytometry experiment. I'll put a link to this in the description below, but this document is perfect for learning what you need to publish in your flow cytometry papers. It goes through step by step everything to include in each of these categories, so you can use it as a checklist to make sure everything you need for your experiment is reported in your papers. And this was put together by the ISAC, or the International Society for Advancement of Cytometry Data Standards Task Force. It is a really great resource and I highly recommend you use it. Now that we've gone through the basics of what you should include in your publications, here's 10 tips to make your data look great. Number 1, always check your run stability. So this goes back to our initial fluidics lecture, where we looked at normal stable flow versus improper fluidics. Now you can either do this manually and gate around problem areas, or if using FlowJo, there are a number of plugins that will automatically remove these for you. I would recommend taking a look at those as they will really simplify this, especially if you have bad fluidics. Tip number 2, remove your doublets. Again, going through a proper gating hierarchy and ensuring that your doublets are removed from your analysis will always result in better data. Again, this can be done simply by using forward scatter area versus forward scatter height and looking for that shift in the population that occurs when you have doublets. Tip number 3, use backgating to your advantage. Backgating is a feature that you can find in FlowJo that will essentially take your population of interest and show where it falls in previous gates. So this will allow you to assess if you've missed any of your cells of interest, but also to show that your cells are showing up where you would expect them to on your previous plots. It's also really great for finding any problems you might have with your reagents or your workflow. Tip number 4, use bi-exponential or logical display. A lot of the times you may find part of the background signal stuck along the axis. A bi-exponential graph will allow you to pull these then off of the axis and allow you to then see the whole population. This is also really good for assessing if you're compensated properly, because a lot of the times those overcompensated populations will lean into the axis and you may not see them. So switching to a bi-exponential display will not only make your data look better, it will also allow you to highlight potential problems and go back and correct them. Tip number 5, use large dots, or better yet, a contour plot. So if you don't have a lot of events on your plot, it can be sometimes hard to see the small dots. If you want to stick with the pseudo color or the dot plots, then I recommend in FlowJo switching these to a larger dot so that they're easier to see. Contour plots though can also really help in this case, so I'd recommend taking a look at both and see which presentation format you prefer. Tip number 6, if using histograms, offset them so they're easier to read. You'll notice in the top image, when you have a lot of histograms on top of each other, it's really hard to determine where the peaks are. By offsetting the histograms, you can then see each individually while also still comparing where they overlap. It's really powerful and makes these overlays much easier to read. Tip number 7, back to contour plots. Contour plots are fantastic. They work well when you have few events. They work even better when you have many events. A lot of the times when you have dot plots and you have a lot of events on that plot, the populations will start to all merge together just due to a natural spread of the population. A contour plot allows you to then still pull these differences out while maintaining all your data on the plot. Tip number 8, always use median to look at fluorescence intensity. In many papers, you may see MFI presented. If you're using this, always make sure you define what the M is, but in your experiments, the M should always be median, not mean. When working on a logarithmic scale, mean is not going to be representative. It will be very subject to outliers on either side. Median or geometric mean are much preferred to mean when looking at fluorescence intensity on a logarithmic scale. Tip number 9, include the number of events on the plot somewhere on the plot. This is especially important if you're using a contour plot. With dot plots, readers can have a fairly good idea of how many events may be on that plot, but it's pretty difficult to assess. Knowing this number will really aid them in interpreting the data and also understanding the precision of the analysis. And last but not least, always look at your n by n plots, or a multigraph display of every marker displayed against every other marker. We talked about this previously in the compensation video, and this also applies here. So yes, you can still use it to look for compensation errors. You can also use it to look for interesting populations that you may not have considered in your gating structure, and may actually find something interesting. So those n by n plots are critical in every experiment. And last but not least, it's always important to recognize the use of a core facility somewhere in your manuscript. These publications are a key metric used to grow the shared resource and bring in things like funding and instrumentation, which in turn makes the core better for you and your future research. So remember to include this in every paper you publish. If you're wondering how to format this, I've shared a sample acknowledgement in the description below. And that brings us formally to the end of our Introduction to Flow Cytometry series. Thanks for sticking with us through the entire journey, and we hope you stay tuned for exciting things coming to this channel in the future. So you don't miss them, make sure you've subscribed and click that little bell icon so you know when the next exciting episode comes out. And with that, we'll see you soon.